It's 12:30. Maybe we should go ahead and get started.
All right, everyone. We'll go ahead and get started now. Hello and welcome to the first session of our evolution seminar series for this semester. Since it's the first one, I'll just briefly say our speaker will go until 1:30, leaving as much time for questions as they like, at which point they will have their private session, a Q&A session with the undergraduate class that's attending. If you're in that undergraduate class, I'm not going to tell you can't ask questions during that section, but maybe just wait and allow people who aren't to ask questions if they'd like to. So I'm really excited to introduce today's speaker. Professor Vatsan Raman is joining us from the Department of Biochemistry here at UW-Madison. Has come to talk to us about a particularly fascinating and useful type of virus called bacteriophages. So without further ado, let's get to the fun part. Please join me in welcoming Professor Raman.
Thank you. And it's a real pleasure to be here. I have more slides that I can fit in an hour-long presentation. So we'll just go as far as we get. The more interactive this is, the more fun this will be for all of us. So really feel free to interrupt me with questions and ask me as many questions as you want. All right. So, I'll sort of provide a general overview of the way we think about science in my lab. And then we work with these high-throughput approaches to study a variety of different systems. I'll focus on bacteriophages for today's presentations, but maybe the first 5-7 minutes, I'll give you a sense of how we think about large-scale sequence function relationships in biological systems. All right. So this is an almost current version of my lab. And so these are people who do all the cool stuff. And really propel the science forward in my lab, and it's an honor and a privilege to be their advisor, and I get to give this presentation. Okay, all right. So as a biochemist and a bioengineer, our goal is to really figure out the molecular rules of life, and to achieve this goal, we sort of pose two types of questions. Right, one question is that of mechanism, which is how different biomolecules interact with each other to carry out some function, because of transcription, translation, and all that. The other kind of question that we pose is, what is the molecular basis of function? Which is, if you imagine sort of a protein or an RNA, what is the contribution of every amino acid in that protein or an RNA molecule towards its function, right? So, because the collective output of that is of a protein is the function of each amino acid within that protein. And I'd argue that over time we've gotten really good at figuring out mechanism. We're good at wielding a scalpel to sort of peel off layers and understand mechanisms at some level of detail. We've also gotten really good at understanding the molecular basis of function, and structural biology tools have played a major part in helping us figure out the molecular basis of function. And yet I'd argue that there are sort of major gaps in our knowledge. And the reason for that is, structural biology tools will give you a structure, right? And going from a structure to molecular basis of function, that's not a straight path, right? And so we can pause here for a second and ask, well, what do I mean by molecular basis of function? And so again, if you imagine, sort of a simple protein molecule, the overall function of the protein is really the collective contribution of every amino acid in the protein, right? So when I say something like, so what is the role of each amino acid towards, say, its specificity, its activity towards a substrate, stability, affinity to other biomolecules, allostery. And so this is the molecular basis of function. And so the way we think about this question is, if you want to figure out the molecular basis of protein function, we have to ask how sequence variations impact protein function, right? You cannot just look at a static protein sequence and impute function from that. You have to introduce sequence variations and ask how these changes impact function. Now, this really gets to the point of this idea of what's sort of the bigness, quote unquote, of protein sequence space, right? And what do I mean by that, right? So imagine, a simple, 100 amino acid protein. If you want to explore the protein sequence space around this, let's say you want to introduce all 19 non-wild type positions at a given site, right? And if you want to do this across all 100 amino acids, that's 1900 mutations, right? Now, this is a very large set of mutants to process for, say, a traditional biochemistry or a traditional genetics lab, right? Now, if you just take it a step further, you look at sort of all combinations of double site mutations. Now, this is a million mutants, which is way beyond what anything we can experimentally assayed using traditional approaches, and then you can sort of take it a step further, right? Now, nature, of course, it samples all mutations at all positions all the time, right? So it does this fairly exhaustively. And so if we really want to figure out how these sequence variations impact function, we ought to be able to make functional measurements, which are not the scale of what we do now, which is say 10 to the 1, 10 to the 2 mutants in an arrayed fashion. And we want to be able to scale this up to say 10 to the 6, 10 to the 9 mutations, right? So the ability to make measurements at the scale at which nature samples them is absolutely essential to map out the sequence function landscape. And so that's sort of one way of sort of asking this question is, if you imagine a sort of this fictitious sequence function landscape, on the y-axis, you have some measure of function, right? Some activity, stability, whatnot. On the x-axis, you have some measure of frequency. And so some measure of frequency. And so what we do now, right? So the current paradigm is, we pick, one sequence, the wild type, one or a few other mutations, right? A couple of more sequences. And what we really see is a small sliver of the sequence function landscape, right? And so the example that sort of resonates in my mind is this idea of these 5 blind men, right? And so these 5 blind men, they touch different parts of the elephant, and they collectively have no clue what the elephant looks like, right? And so as we, this is akin to what we do now, right? We pick, one or a few mutations to a protein or even a genome, right? And then we say, well, we have no idea how this biomolecular system functions. Now let's imagine instead of 5 blind men, you have 50 blind men, right? And now they sort of collectively have a slightly better sense, right? And again, as you start sampling more and more mutations, right, you start seeing the peaks and the valleys in the sequence function landscape, right? And slowly the protein starts revealing itself to you. And you can take it to an extreme and say, well, now I have 5,000 blind or 15,000 blind. And so again, if you start measuring function at that scale, the entire sequence function landscape reveals itself. And so this is sort of a molecular blueprint, right? It tells you how these changes impact function. And then again, you can use that information to impute mechanisms. You can impute function. So that's kind of what we do. And then the question is, how do we measure function in high throughput? I mean, really the trick is to be able to link the activity of that protein to high throughput sequencing, or link the activity of a genome to high throughput sequencing. And by doing that, what we do is, if you can sort of take every experiment, some of you may have done genome sequencing or sort of whole genome sequencing, you get billions of reads, right? So if you can turn each read into an output of function for your particular biomolecular system, well, then the number of measurements you can make sort of trivially scales with the sequencing volume, right? And so really the trick here is to establish that link between the activity of your biomolecular system and high-throughput sequencing. And so much of what we do in my lab is really figuring out how do we establish that connection for an arbitrary function, right? And then how do you make the connection between activity and high-throughput sequencing? There are a few different ways one can go about this. This is simple fitness is sort of one example where your protein or interest is essential for cell survival. That's why you mutate it, and then you kill its activity, the cell survives or does not survive. And so that's sort of a trivial way in which you can link the activity of a protein to high-throughput sequencing. There are slightly more sophisticated ways where you can maybe have a reporter gene, which is barcoded, and then again, you can measure the number of transcripts, and so that's another way of doing it. Or you can use sort of single cell measurements where each protein, a perturbation that you introduce to a cell, can be measured as sort of this large n-dimensional transcriptomic state. And again, there's variations within that. And so we do all of the above. And so what I've sort of described to you is how we can systematically introduce gene variations to single gene, right? And this concept then can extend to whole cells, right? So you can say, well, instead of a single gene, you can treat the entire genome as sort of a single entity, and the removal, sort of deletion of individual genes as perturbations, and ask how does deleting a certain gene impact its function? And so this is sort of akin to sort of traditional CRISPR screens that people have done. And so far I told you how we can use these approaches to understand biology, and then flip this question and ask how can we use these approaches to engineer biology? Again, once you get these large sequence function landscape, it tells you how changes can impact function, and then you can basically use that information to engineer proteins that have interesting properties. And so again, it's a protein scale, the genome scale, it's the same concept. Instead of looking at individual amino acids, you can look at entire genes. It's exactly the same. And so we've sort of taken this general paradigm and we've applied this to a variety of biological problems. And not just to sort of looking at proteins and genes, we've been sort of thinking about designing inhibitors or agonists or antagonists to different sort of cell surface proteins. And again, the idea there is to introduce a number of perturbations and then be able to measure function. So you can ask if a certain, say, peptide, designed peptide can shut off a cell receptor or activate a cell surface receptor. And so again, we've been thinking about this in a few different ways. We've been looking at biological sensing. How can we design or understand sort of proteins that sense different molecules? These nanomachines are these bacteriophages, which are sort of really cool entities that one can use both as a sort of a fundamental study of protein sort of biological evolution, but also as agents that can be employed to treat multi-drug resistance. And sort of a few other systems that we're working. So, but what I'll do today is just focus on our work with bacteriophages. And again, what got us sort of thinking about this is this sort of big looming question. Well, it's not even a looming question. I mean, we have the problem now. It's sort of drug-resistant bacterial infections. This is a huge sort of slow-moving pandemic, if you will. And if you sort of look at the CDC's worst offender list of these drug-resistant strains, some of these strains are resistant to 15, 20, 30 antibiotics. So it's a really quite lethal bacteria that are prevalent in our environments. And there's also this large class of these microbiome-linked diseases, where microbiome is a complex community that lives within our gut and other environments. And there are certain bad actors that might induce dysbiosis. And how can we precisely change the composition of the microbiome to remove the bad actors but keep the good ones? So these are just some of the motivating questions. Let's get this. Let's get these slides. Yeah, so the projections are that antimicrobial resistance is likely to be the largest killer in the next couple of decades if we don't address this issue. The traditional small molecule antibiotic pipeline is basically running blank, right? So A, there aren't that many molecules that are being discovered. B, those that are being discovered, there is spontaneous resistance that emerges. And C, even if you are super successful in identifying a small molecule that does not induce spontaneous resistance, basically the clinicians will put that away as a drug of last resort because they don't want to use that molecule, right? So you go through all this trouble to identify something that works, and the clinician will basically bank it. And so what we wanted to do was we said, well, how about we sort of go back to the drawing board and see how nature solves this problem, right? So, these are bacteriophages are really cool entities. There's the largest biological entity on Earth. This is an evolutionary biology seminar, right? You can think of it as the longest evolutionary experiment on Earth, where the phages and bacteria are constantly trying to outcompete each other. And so this has been the idea of using phages as therapy is not new. People have been thinking about this for well over a century. And this is an image of this place called bacteriophage in Paris, where they sold these cocktails of different bacteriophages for all kinds of different conditions, colitis and all that. And this initial avatar of this place eventually then became L'Oreal, which is now the cosmetic company that we have. So its origins were actually as a bacteriophage company about 80, 90 years back. And so, just sort of biology 101, the way that phages work is it absorbs itself. So this is a bacteriophage, sits on a bacterium, delivers its DNA, and then it hacks the machinery of the host phage, of the host cell, makes many progeny baby phages, and then these phages then pop the cell and they go on. So this is a lipid life cycle. There's a different life cycle where it integrates the genome, but we'll sort of ignore that for a second, right? And so in sort of some ways, you can think of these phages as sort of the ultimate smart drug for a few different reasons. One is that they're extremely precise, right? So these phages, they target a specific strain or a couple of different strains. And so that gives you a level of precision that antibiotics simply don't have, right? And especially from a microbiome disease standpoint, if you want to remove the bad guy but keep the good guys in place, this is a level of precision that's kind of important. Two, they're evolvable, which means that when bacteria become resistant to a bacteriophage, which they almost certainly will become, right? You don't have to go back to the drawing board. You can sort of re-evolve the bacteriophage to be a few steps ahead of that. This is a cool property. It's auto-dosing, which means it kind of amplifies at the site of infection. Right? And four, it's the abundance, right? So it's just basically an unlimited supply of bacteriophages that one can choose. If there was sort of one example of this couple where Patterson had this Acinetobacter baumannii infection. And then he was treated with bacteriophage on compassionate grounds, right? So, there is no approved phage therapy in the market by the FDA, but you can request the FDA to do sort of compassionate therapy. And he was treated, and it turned out to be a big success, and to really sort of galvanize the field to go back and ask if phage therapy could indeed be a viable solution. But you got to ask if it's, if it has all these positive attributes, why don't we really have phage therapy, right? It's, what's holding us back, right? And I'd say there are a couple of fundamental challenges with the use of natural phages for therapy. One is they have narrow host range, right? Now, I just sort of played that up as a positive attribute 2 slides back. It also turns out to be a negative attribute, because if there are strain variations of a given pathogen, and if you have a strain variation of a pathogen and I have something different, then we cannot customize phages for that, right? That sort of approach simply does not scale, right? That's number one. Number two is the efficacy of natural phages kind of sucks, right? And the reason for that is if you're a phage that is extremely good at killing your host, well, you're going to simply perish, right? You don't have a host, right? And so there is this evolutionary selection for mediocrity that's happened with natural phages. Third is that these natural phages are often used as these phage cocktails, where you have multiple phages put together. And often these are highly unrelated phages, right? So who knows what their life cycles are, and then what their mutual interactions are like. And so you just have this inconsistent results that just simply won't be palatable to, say, for FDA, right? Because they want to have batch-to-batch consistency every time you deliver a treatment. And bacterial resistance, this is a huge part, right? I mean, so bacteria will become resistant to a phage. And the question is, do we have methods in place where we can always remain a few steps ahead of the bacteria, or if we can preemptively block off pathways of resistance? And so I'll give you sort of one example of what we've been working on. So this is, the phage, which is interacting with the host receptor. And so this is the receptor binding protein, which is the primary point of contact between the phage and the host bacterium. And so you can imagine that it basically drives the activity in some ways. How well does it stick to its host receptor? And B, the specificity, which is what are the different hosts that it can attach to, right? So it's sort of a governing, it governs both of these properties of the phages. And so this is a very prototypical bacteriophage T7. People have studied this to death for the last 50 years. This is the receptor binding protein. Here is the very tip, so we'll think of it as a shoe, right? And this little domain has a major role in both the ability to absorb on a bacterial receptor and the types of different bacteria that it can bind. And so we said, well, what if we do this sort of mutational experiment where we systematically mutate every amino acid of the receptor binding protein to all remaining 19 non-wild type positions, right? So it's an 83 amino acid proteins times 19, about 1,600 different. So sort of relatively small library. Well, the problem was that we simply did not have a method to do targeted phage genome engineering where we systematically replaced genes at a given phage locus with variants of interest. And that was the work that Phil Haas in my lab did, where he devised a method where he could introduce a cassette of containing a library of different variants at any targeted site of the phage genome. And what makes phage engineering particularly challenging is you have to introduce this donor cassette through the host bacteria as the phage is actively killing the bacteria, right? So on the one hand, the bacteria is basically dying. On the other hand, it also has to deliver this donor cassette to your targets. And so there's a lot of optimization that Phil had to go through to get this to work. It's called Oracle. I'm going to just skip through the details. Basically, it's a high-throughput locus-specific phage genome engineering method that helps us create large unbiased libraries of phage variants at any target. Let's just skip through all this. We essentially have an oligonucleotide pool where we introduce variants of a given gene. We can have these companies that make this pool for you. And so this pool contains about 1,600 different variants, so sort of printed DNA. And you can introduce variants of interest. And these variants would then be used, would be substrates for a recombinase that would insert them at a given site. Right? And so now you have this population of variants. I'm going to skip through this all the method. And so the way this would look is as follows. So let's say this is amino acid 1 through 83. You would have 19 sequence variants at position number 1. You have another 19 sequence variant at position number 2. blah, 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 all the way up to 83, and you sum them all up with the wild type allele, and then you have a 1577 variants, right? So how does the selection work? The selection is super trivial. So if you have a phage variant that is deleterious to phage infection, if you apply it on a target host, basically it drops out selection, right? And if you have a phage variant that has no impact on fitness, the abundance before and after selection will be more or less identical. And if you have a phage variant that has higher fitness, right, then you have gain of function. I'll just call it higher fitness. Then you have more progeny, right? So what you can do is you can simply count the number of variants after selection, number of variants before selection, and then sort of determine a ratio, and that gives you a fitness score for a given variant. And so let's I'll give you a sense of what this data looks like for a couple of positions, and then we can look at the whole heat map. And so this is position number 1, and what you'll see is, so this is glutamine, and so you have the Q here, wild type position. The red means the intensity of red is activity level. So you can see that a couple of, and white is lack of function. And so you see the 3 positions here or 4 positions that are these mutations completely kill activity. The vast majority of mutations are still functional, but what you already see is there are a couple of positions that are more red than the wild type Q, right? So right off the bat, what we have is we've done slightly better than what nature has already done in terms of activity of this P7 on its host. So this is position number 1, right? Now you can look at position number 2, and you kind of get a completely different story, right? You see that most of the variants kind of drop out of selection. There are a couple of conservative mutations, alanine, valine, and isoleucine, that are close to the wild type position. Those survive selection, the rest drop out of selection. And now you can sort of look at the whole map. So those are 2 positions, and now we can look at all 83 positions in a sort of to get the complete sequence functional landscape. And just a couple of things to point out. Again, more red is more activity. The position with the black box in the little on the corner, that's the wild type amino acid at that position. The white kind of drops our selection. So that's what this is. So this is the C-terminal end of the receptor binding domain, which is floppy. And so any substitution there should have no impact on function. And we kind of see that, right? So you want to go through these few checks that, it's not garbage in, garbage out. So you have to have the sanity check to make sure that the data that you get matches your either known prior biochemical or genetic knowledge, or even your intuition of how these systems should. So this is a floppy little bit at the end. So really no substitution should matter, and see that. What we also see is that, the vast majority of these mutations are deleterious, which is perhaps to be expected. And so the fitness peaks, so if you sort of imagine this again as a sequence function landscape, there is sort of the sparse fitness peaks in this space. But what's really cool again is that now we have phage variants that are, these sharp red patches, which are about 20 times, 25 times fitter than wild type on a given, right? And again, so think about it from an evolutionary standpoint. This is a T7 E. coli pair that have fought each other for, maybe hundreds of millions of years, right? And again, a simple mutational scan already reveals itself as mutations that are better. Now, it's important to note that this is just one host. So again, for a wild type T7, there will be other trade-offs that it has to keep in mind, right? So one argument could be that as you gain fitness on a given host, perhaps you lose fitness on a different host, right? And so if you're the wild type phage, you have to maintain this balance to be able to be nimble-footed enough to be able to infect a different host, just depending on which environment you find yourself in. Now, the other cool thing is now we can identify these regions which are rich with mutations that enhance function. Now, if I sort of put on my bioengineer hat, I'd say, well, this is kind of cool because now if I want to go back and mutagenize or make combinatorial mutations, I don't have to waste all my time on these regions. I can just focus my effort on regions that's going to be most fruitful. Right. Now, what's really handy with these pool screening experiments is once you build the library, you can sort of keep dipping it in different sauces and you get different results, right? So you can take the pool of phage variants, you can infect it with strain A, strain B, and strain C, and you get different maps. And each map kind of tells you it's sort of a unique signature of the interaction of that phage with that particular phage, right? And you can then take all that information, collate them, and ask, If I now slap that information across different strains on the simple domain, right, which is sort of, as simple as it gets in terms of structural biology, right? Then you can ask, well, which positions are positions that are important for function, which positions are important for stability, which positions are important for specificity, and that's the kind of information that we can do with these maps of universe, right? And so these blue positions are the positions that have higher activity towards a specific host, and are the positions that drive specificity towards different hosts. We can take this a step further. While we know that these are strains that were naturally susceptible, right? What if we delete the native receptor, right? Can we still have a variant that survives? And so this is akin to an experiment where we have a resistant, right? So one way in which a bacteria might become resistant to a phage simply stops expressing the receptor, right? And so we can simulate that condition here where we delete the receptor and now challenge the library again and ask if we have a gain of function. And so this is the lipopolysaccharide is the native host, the receptor of T7 and E. coli. We can delete the genes, the biosynthetic genes that make the lipopolysaccharide. So this is a funny plot, but it took me a little while to figure this out. So closer to 0 is higher activity, more negative means it's poor activity, right? So you're looking inverted. I've asked a student to flip this around, but he's adamant that he won't do it. So anyway, which is a lot better. So this is wild type E. coli infected by T7, high activity. The moment you delete the native receptor, right, there's about nearly 6 log loss of activity, right? And so we challenge the pool, right? The same deletion variant. Again, we find these sort of bright red positions, which are potentially gain of function. We can now isolate these individual variants and now apply them on the specific deletion and in a sort of clonal level and ask, do you get gain of function? And now we actually have multiple variants that can both kill the wild type E. coli and kill the deletion variant. Again, so you can sort of think of this as a simulation of an experiment where the host acquires resistance by turning off its host receptor, right? But now we have, the advantage of having the pool is that even if the wild type misses it, there is already somebody else who's ready to sort of hit the bacteria from a different direction. And this is an important point because as we think about multi-drug resistant mutants, they'll have many, many mutational pathways by which they can break free from the selection. And you want to have variants in the pool that can block off those pathways. So we said, well, this is all fine. We've done this with the lab strains. Now, really, let's take it to the bad guys, right? So we looked at the CDC list. And so this Enterobacterellus, this is the NDM1 superbug, which is resistant to about 30-35 antibiotics. We actually did this in the lab. I stayed away from that experiment as far as I could because it's a really, as sort of dangerous a strain as we can have, and susceptible to 2 antibiotics. So we took the strain. It's a growth curve. I want to sort of walk you through what this is. I couldn't get the animation working. Now, if you apply, so this is this is simple optical density curve, right? So the black is the no phage challenge, right? So it just grows fine, right? So this is the Enterobacterellus strain. If you apply the wild type phage, sorry, the gray is the no phage, right? If you apply the wild type T7 phage, really has no impact, it's just fine. If you apply the initial library of mutants that we find, right, the single amino acid mutations that we find, it's actually dead for a few hours and some resistant mutant emerges. And then it kind of takes over the population, but we're able to, it remains dead for a few hours. Now, what's nice about this curve is now we can sequence the phages at an early time point, and we know which phages were enriched that could confer that ability to kill the strain, right? Now we can take that information and start making another combinatorial cocktail. That's, so imagine this as second generation cocktail. And then we get this little bump, but now this is dead for 15, 16 hours. So this is kind of promising that we can now iteratively improve upon these phages. And sort of going back to the earlier argument, how do we stay a few steps ahead of these multi-drug resistant bacteria is to be able to simulate these pathways of resistance. So you basically want to let them acquire resistance every possible way, and you want to ask, can I preemptively block off those pathways? So when you have a final cocktail, it would not just be effective against the wild type multi-drug resistant strain, but would essentially contain solutions against all possible pathways of resistance. Okay, So this is fine now. This is trigger warning. We're going to look at a couple of gory mouse pictures. So feel free to look someplace else if you don't want to see it. So we said, okay, we've done this in the lab dish. Now let's actually do a mouse experiment and see, does it work, right? So it's in vitro is fine. And so what Phil did was he established what's called as an abscess model. Just forget about the text. I'll just sort of tell you what it is. The abscess model is where we deliver the enterobacteria to the back of the mouse, and you just have to deliver at the right concentration so that it forms a sustained infection. And if you leave it untreated, the mouse basically dies in about 5 or 6 days. If you overdo it, the mouse will die quicker, but you sort of want to have it about the right time so that you can provide a potential treatment or a control and see what happens, right? This is all just the details of the abscess model. Already showed you this information. And so we had sort of 3 different conditions. One is just the saline treatment, right? There's no treatment there. The wild type phage. And then we had this combinatorial library, where we had about 10,000 different members, which were from the 2nd generation. So do an initial mutational scan, get the ones that appear to work well, make some combinations thereof, and we have about kind of the 5 different variants, right? And so this is at the end of, these are not marked, but I'll tell you what they are. So this is the abscess at the end of day 6 or 7. This is a big gaping hole in the mouse. So this is wild type. So this is the saline treatment. This basically infected all the organs inside. The mouse is basically dead at this point. This is the wild type T7 treatment. Again, just about the same as the saline treatment, sort of a large, gigantic abscess. And these are the 3 mice that have been treated with the common protein, right? And it's, I mean, there's still a little bit that's left, but by and large, the infection is gone. So this to us, we were very excited to see these results, that what we did in sort of a laboratory setting could actually be translated into an animal experiment. And now we can make sort of other measurements that quantify this. So this is the survival curve. The orange is the library that we introduced. Again, this is an N of 6 experiment. All the 6 mice survived. The saline treatment, about 2/3 died, and about the wild type phage, about half the mice died. Again, this is one pathogen, and this is one phage. So what again is promising for us is that in principle we can take the same approach, apply them to other classes of pathogens and passively we can do that. So the other sort of summary stats. This is the abscess size. This is the abscess size with the saline treatment, abscess size with natural phages, and this is the abscess size with engineered phages, right? And here is the bacterial load. So we had about 10 to the 9 bacteria that we initially introduced in the challenge. At the time point at which this was measured, the saline and the natural phages had about 10 to the 8, 10 to the 9 bacteria. About 10,000-fold drop in bacterial load with the engineered phages. So we felt like, again, we're barking up the right tree here. And so we want to move on and try other pathogens. So where are we? Just to go back to the point that I made earlier. The one issue was with these natural phages was narrow post range. And I think at this point we have methods in place where we can take natural phages, make changes to them. There's this whole machine learning angle that I've not described, where we can take these large data sets of sequence function measurements, apply them with these large language models that people have been developing with proteins, and then now we can basically give the phage, or give the model any desired objective, which is say, infect this, this, and this, but not this and this, and it'll be able to predict sequences that have those problems. And so we're at the point where we think we can start programming the post range. We can introduce mutations that remove these evolutionary obstacles, right? So we can take the mediocre phages and make them highly potent. Instead of starting out with these inconsistent phage cocktails that were assembled and not with any particular rationale, we now create these variant pools, which are variants of a single or a couple of phages. And the idea is that now we have all potential pathways of resistance kind of locked off. I haven't had a chance to describe what this is about. So we've started screens where we can identify these anti-phage mechanisms. So bacteria have a plethora of anti-phage mechanisms that they use. So restriction modification is one of them. CRISPR-Cas9 is probably the most famous one. But then phages have evolved counter strategies against these resistance, these phage defense mechanisms. And so we've been screening the metagenomes from bacteriophage genomes to identify which counter strategies are shared across phages, and if we can essentially copy paste those strategies into our therapeutic. So at this point, I'm going to stop. I have another 15, 20 minutes of material, but that's on us taking off in a different angle. But perhaps this might be a good point to pause and have Q&A. What was that?
You've got time.
But we're going to transition to a different direction. So, but
Okay.
I'll just let people ask me questions.
Just wanted to make sure that you weren't stopping.
Yeah. What is the time?
It's 1:05.
Yeah. I'll.
The screens that you do for the activity of these bacteriophages is in vitro, right? And some of the applications that you're anticipating potentially, notwithstanding the one you showed us, are in the gut. Right,
In the gut, you said?
In the gut,
Yes.
yeah. And so, but you're not replicating the milieu of the gut, and that's kind of hard to do. But one thought I have is that there are molecules in the gut that really do affect protein structure and so on, like bile acids, which are very abundant in the gut. And I just wonder if you could take advantage of that, because you mentioned earlier that there's a selection for mediocrity in vivo because you're killing off the host. But if you could devise a screen where you're selecting for mediocrity in vitro that is then converted into much more success in the milieu of the gut. So for example, you could look for, you could do a screen with, taurocholate, just as an example, but maybe it's a cocktail that mimics the milieu. And if you could find bacteriophages that are mediocre without that, and then you put them in the gut and suddenly they're incredibly good, wouldn't that be an advance?
What would be the advantage of having that combinatorial effect with a small molecule? Why not just have potent phages against the microbiome target to begin with?
Well, because you have a means of replicating it.
But I mean, we can run these screens in norabiotic mouse gut or mice gut, right? So we can take sort of introduce human gut samples into them, let them have stable colonies. And so one experiment that we haven't done yet, but we're going to do in the next month or so, is we have these certain E. coli that are that we find in food infections, and so we can specifically introduce that into your prototypical gut, right? So essentially, we've created this infection in the gut, right? So you can have the milieu of the gut, but with this pathogen present. We challenge it with our variants and ask, can we precisely remove the pathogen but leave? So we can do a 16S sequencing before and after selection and ask, are we specifically depleting the pathogen without touching the others? But we can do this in the gut milieu. It doesn't. We can certainly do a baby version of that screen in an in vitro setting, right? Just to get a sense of what potential candidates we want to use. But once we have that, we can just sort of run that in the gut. Does that make sense?
It does. So then the question is, you can answer if my premise is right? In other words, you've already done the experiment. So when you do that and you compare your screening results in a purified in vitro setting versus the milieu of the gut, are the results very different?
So in this case, so the one thing that I'll give you an example of this abscess model, what we have not done is we have not screened the phages that came through successfully in the abscess model. In other words, since the conditions are different, it's a tissue environment with all kinds of other things, right? So we don't know if the best hits that we found in the in vitro screen are the same or different from the in vivo screen, right? My prediction would be that they're likely different because you have to optimize for just the ability to be stable in that environment, right? Maybe perfuse wherever it has to go. So there are other physical chemical barriers that the variants have to sort of meet those challenges on top of being able to kill the bacteria. So I think there's just sort of multiple layers of selection, which is much more complicated in an in vivo setting that's not accurately captured in our simple dish schemes. More questions? Yeah, John.
Curious how you would imagine this ultimately being applied in a widespread clinical context. Are you thinking that it would start when, if someone has a drug-resistant infection, would it start by sequencing, whatever's infecting them, and then assessing whether this work has already been done for that strain? And if not, it seems like there's going to have to be some kind of ongoing efforts because the bacterial strains will continue to evolve, things that can resist this too, even though they're kind of blocking their first evolutionary step within a particular host. Is that the sort of system you're imagining?
Yeah, so I'll give you sort of the grandiose vision and we'll sort of bring it down to earth, right? So the idea would be to have a universal cocktail, right? So let's say, it's like an 80-20 problem, right? Which is 80% of infections are probably caused by like 6-7 different strains, right? Or 10 different strains, right? So if you can have a universal solution against that set, then what you would say is, as long as I know you have a bacterial infection, my first line of treatment would be what this universal cocktail. And so these would be variants from multiple pages that we've sort of pre-curated based on their activity against, say, multiple pathogens. So just sort of this combinatorial set. Now, that might get you some fraction of bacterial infection, but maybe others where it's simply not the solution simply doesn't exist in the phage population that we have, and we'll have to start looking elsewhere. And so then the next step would be, we can start creating this bank where we have a number of clinical isolates, right? You challenge the phage variants against them, and you know which groups of variants are active against which clinical isolates. And so using that information, you can again create this minimal cocktail that is active against, say, let's say Klebsiella infection. You'd say, I have a cocktail of variants that is active against 60, 70, 80% of all Klebsiella clinical isolates, right? And so the next stage would be, I have some knowledge of what the pathogen is, and I can give you a more customized treatment, right? And so I think we'll sort of have to work it that way, where the ideal goal would be that this, just with an antibiotic, you maybe don't need to know what the pathogen is. You provide the treatment upfront, and if that doesn't work, you have a plan B and you have a plan C. So the idea would be to have, one or a few different universal cocktails and see if it works. And if it doesn't, then you start gathering more information about the pathogen and then maybe give a more customized solution. I guess that's the way I'm thinking about it. Yeah, go ahead.
Me?
Yes.
Yes. Do you mind putting the graph with the curves, the growth curves on it again? Because I realize I don't quite understand, this one. So if I had to, we have one where it was and labeled. So the red one was the original cocktail of single mutants.
Yeah.
And when you said the blue one was you took first, 4 hours, the ones that were there, and you amplified those.
And so what we do is, once we know, there are some potential solutions over here, right? So we can take those sequences just based on so we know what those sequences are based on deep sequencing. We can start making combinations of those because it's a single mutant. You can start making combinations of 2 or 3 or 4, and then come up with the second generation cocktail, right?
I see. So it's not just enriching for the ones that were there, but it's generating recombinants among them.
Yes, you're sort of
adding some sex in there.
Right. So you sort of like a sort of traditional DNA shuffling experiment using a set of templates that have that come out of the screen here.
That explains why it's a little worse initially, but why it sustains.
Well, I have no explanation for this. I don't know why it happens, but it is what it is, but it actually does. It could be that maybe we were missing some critical variants in that set, and so maybe there was an initial burst of activity. But I mean, it'll be interesting to sequence here, but we haven't done that yet.
And if you take did you sequence the ones from the very end?
No, we have not done that.
Yeah, because that would be nice to see if they would just be great.
Correct. Yes, exactly. So you want to take the hits that come out of this, reapply, and there should be a flat line all the way through. We haven't done that. But Brian, I don't know if you have a question.
So the Uber cocktail that comes in at the first, is that every amino acid at every position?
Yes.
So it's not only huge complexity of a single page, but it's all the other pages that you put into this cocktail, the
It's-
same diversity.
It's one page, but we made 1,700 variants of that page, right? Of that particular receptor binding protein, each, I go back. So this is a population of pages, where it's one, each page contains one mutant at that receptor binding protein. So this has a different mutant, this has a different mutant, this has a different mutant, likewise, these are different, right? But collectively, there are 1,700 variants in one pot.
So it's not an issue of multiple mutations in these pages at this point.
At this point, it's, in principle, it's just one mutation per page. In reality, you know, pages mutate, and there might be other mutations, but in principle, there should be only one. Someone else had a question, yes.
I was curious if you, when you deleted RFAD to make E. coli without LPS and you found T7 that could infect, do you have any idea what the receptor is that they're using?
Yeah, you know, the people have speculated that, you know, ARM-C or ARM-B could be an alternative receptor. We could delete that and find out, but we haven't done that, but that's just speculation. Yes.
You're basically substituting amino acids and kind of like doing phage evolution. So I was wondering when you do that, do you ever encounter any metastatic interactions that kind of makes the evolutionary path of the phage unpredictable, or is it different sensitivity?
Well, at this point, these are only singles. So the more comprehensive experiment would be that you start making combinations of 2s and 3s and 4s and sort of ask if the summation of the individuals and the summation of the complex combinations, if they're additive or non-additive. But you know, we haven't quite done that experiment. I'd imagine there is some epistasis that almost certainly has to be the case. The question is one of degree, right? Is it rampant or is it sparse? My guess would be for a small protein such as this, it's probably sparse, but that's just a guess.
You have a lot of these mutation scanning experiments, you have data every point for every amino acid, so you can get really fine scale information about the distribution of fitness effects.
Correct.
And if you look at different parts of the protein, you see, do patterns come out like certain chemical properties of amino acids that are retain fitness in the structural part of the protein? Do you have a DFT for that? And do you have a
Yeah, so I'll give you an unsatisfying answer to that question, which is we stared hard and tried to make sense of it. And we could, well, actually, there's one exception, I'll point that out in a second, which is, so these positions, the ones that have high infectivity, are all arginines, histidines, and lysines, right? So this positively charged amino acids, you know, why could that be? It could be maybe the surface of E. coli is negatively charged because of the phosphate head group and whatnot, and so maybe this is like an attractant. But then there are other hosts on which these don't win, right? So, you know, make sense of it, what you will, right? So what we've done, so like I said, I've scattered these maps and we really couldn't make a lot of, well, there's some sort of ground truths that emerge, which is again, the positions in the core of the protein are immutable as, you know, structural biology 101 will tell you that the protein core has to be intact for stability. The other nice thing is these little loops at the end, which are at the very tip or sort of like the fingers, and these are the ones where all the action is. There are certain positions which are on the sides. The blue positions are the specificity determining positions. And so these are likely pointing down, but these are examples where the receptor binding protein, which is a gigantic trimer, you might imagine that this domain in some instances is like this, and in other instances could be flat because then this exposes this amino acid to the surface. But again, it's all speculation because based on where these amino acids are. And so what we've done is we've used this large data set, and you sort of train ML models with them, right? And use those models in a predictive fashion to ask them to generate sequences, and they'll give sequences back. We don't, again, those appear to work really well, but again, we don't know why it just works. Right. And so clearly there is some underlying pattern that's not evident to our observation that the models are picking up. Like I said, it's an unsatisfying answer, but it works. Well, yeah.
To my understanding, excuse me, so you use E. coli and several versions of it, but given a bacteria perhaps with less versions readily available to you for study, do you think that this method would still be effective in creating a phage that could potentially attack that bacteria?
Yeah, so what we're doing now, so there are multiple things that we could be doing, which we are doing that I haven't shown here. One is the single amino acid substitution is like the dumbest version of this library, right? But it's still a useful starting. What we've also done is we've asked, are there sequence motifs that are enriched in phage metagenome? So for instance, are there solutions that phages kind of exchange? Hey, look, this little sequence motif works really well on these large panel of strains. And so there's horizontal gene transfer, but we've started mining the phage metagenomes to ask what these sequences are, and can we copy paste them up, right? So essentially use what evolution has already figured out. So again, there are a number of strategies that we can use from a phage engineering standpoint, which we are doing. But on the other, from the host side, what we're starting to do now is we have about 300-400 different clinical isolates. So these are actual pathogens that have been isolated from people who have the infection, right? So it's not lab strains anymore. These are strains that are real pathogens. And so once we build these libraries, right, we want to apply them on these 100-200 different pathogen panels to ask, what are the common solutions that emerge from this large pathogen, right? So presumably these solutions are the generalists that you really want, right? Or at least it'll give you some inkling of what combinations of mutations you have to start putting together to start making a generalist. So those are the screens. So the initial proof of principle was on laboratory strains and a couple of different CDC strains, but now we have 300-400 different pathogens that we're addressing. More questions?
We got 8 minutes left. Did you have more slides or were you gonna?
Well, 30 minutes of presentation. We can stop here if you want.
Well, I got a question.
Yeah.
So you mentioned the idea of using multiple phage cocktails to not just treat the variant, pathogen variant that's there, but potential resistance mechanisms that could emerge. Considering, since you also mentioned, this is one of the longest evolutionary arms races on the planet, does that hint at some concern for the validity of actually developing those cocktails? Because there's been all this time of this competition between the microbes and the phages, and it hasn't led to the extinction of the microbes. Is the key that you're exposing them to phages that they might not encounter in their natural environment? Like, do you think that suggests that there's a limit to the ability to actually contain every possible avenue of resistance?
It's like the flu vaccine, right? I mean, you keep updating the flu vaccine, right? The virus keeps mutating, you come up with a different vaccine every season. Some version of that is going to apply here, right? It's not a solution that's going to stand the test of time. It'll stand the test of time for a couple of years, and the bacteria will. And the question really is, do we have methods in place where we can always remain a few steps ahead? And I think that's the direction that we're heading. The second point is, the phage host interaction in a natural setting is different from our goal, which is to have complete pathogen clearance, right? Which is you can hit the bacteria with a ton of phages and essentially bring down the load to a point where the immune system can basically do its business, right? The sepsis or other infections just happen if the load is so high that the immune system simply cannot keep up with it, right? So the evolutionary model of phage host sort of competition applies only to a certain extent. The goal of pathogen clearance is a little different in that we just want to knock it down to a point where even if you don't have 99.999% clearance, you just bring it below, say 10 to the 3, 10 to the 4 CFUs, then that's good enough for the immune system to do its business. Yeah.
I also had a question kind of going off of that, because you were talking about kind of not needing to know what the strain of bacteria is that a person is infected with. If you're going to provide them with this phage cocktail, are there concerns then about bacteria developing immunities maybe to the phages that perhaps would give them an advantage? Like you're kind of setting up a natural experiment for bacteria to become like super resistant even to a lot of different phage like variants, or is that a concern?
I mean, like I said, I mean, in principle, it's, I mean, they will learn to become resistant, right? You know, are we gonna make them super resistant? I mean, I don't know the answer to that question, right? I mean, one might imagine that, so one way to maybe think of addressing the question is, there is, again, a limitless supply of phages against a particular bacteria. So even if they evolve resistance against a small panel of phages, which is what the cocktail would represent, right? Then you can go pick a different set of starting phages that the bacteria ought to be susceptible to, right? So I think there are ways in which we can hit them where you, so I don't imagine a scenario where they become broadly resistant to every possible phase that it's the starting points are different. More questions?
All right, let's all give a big round of applause for Professor Ramsey.
Head over to 1360. I also just want to take advantage of this opportunity to remind people to go to evolution.wisc.edu and join the mailing the email list, and also officially join the Institute. Thank you.